Hello everyone, my name is Vipin and I'm here to present you BootTHiC, which is a method that aims to detect transcriptional hubs by integrating HiC and transcriptomic data. Let me first outline the presentation I'll be giving to you. I'll start by giving you some background as to better understand what we mean by transcriptional hubs. I'll then unpack with you what this BootTHiC pipeline really is, methodologically speaking. And then I'll try to present to you some of the results revolving about why is it important to be inside the hubs compared to outside. So first, a bit of background. What do we mean by transcriptional hubs? And this stems from the assumption that the genome is organized as a collection of aggregates that brings different parts of the chromosome in close proximity, thus producing a microenvironment in which we expect to find a particular set of transcription factors, chromatin remodeling factors, and other regulators that ensures that the adequate expression of those particular genome regions is done properly. Such a structure is expected to have two outstanding features. One, an enrichment in chromatin interaction, and especially preferentially self-interaction. And the second one is an enrichment in transcriptional activity. These two features shape how we have designed BootTHiC. Before going into that, I'll be presenting to you some of the technologies that we use as input to the BootTHiC method. First, let me present to you CAGE, which is the transcriptomic side of the method. CAGE is characterized by a high precision, meaning that it's able to have a base pair level precision for the position of the transcriptional start site for the RNA species it's able to detect. This allows it to detect different alternative promoters across samples. The second feature of CAGE is its specificity, more notably its capacity to distinguish between transcription start sites that are associated with promoters, which tend to be biased towards a particular direction for transcription, and enhancer start sites, which are characterized by a transcription that tends to be bidirectional. This capacity to distinguish enhancers and transcription start sites from promoters will be important to keep in mind when I present the rest of the results. The second set of technologies that I'm using in BootTHiC are collectively referred to as chromosome conformation capture sequencing, and in particular here I'll be focusing on HiC. I'll briefly introduce how HiC works. HiC can be understood in terms of a protocol that has four main steps. The first one is a step of fixing the nucleus endogenously inside the cell, and then trying to shear it or cut it as evenly as possible using a restriction enzyme. And then the next step, which is the key step, is a ligation step, which will ligate together pairs of genome regions that are in close spatial proximity by creating a sort of chimeric product. These chimeric products are then sequenced and mapped to produce a long list of pairwise interaction genome-wide. Typically, this long list is represented as an interaction matrix where each axis corresponds to genome coordinates that are typically evenly binned into windows whose size or width reflects the resolution at which we are looking the HiC data at. And this will become important when I present BootTHiC. So now let me present to you what is our actual pipeline. It can be understood in two main parts. The first part revolves around the goal of detecting those aggregates or parts of the chromosome that tend to have a preferential self-interaction. And the way it does it is by first considering multiple resolution of HiC simultaneously. The reason for considering multiple HiC resolutions simultaneously is to capture different kinds of information that different resolutions tend to highlight. Typically at high resolution, we tend to have a very detailed description of the interaction patterns, but at the cost of not being able to capture long-range interactions, which is typically what low resolution HiC is good at capturing. So ideally, we would want to have this particular resolution that is able to get the best of both worlds. So the most level of detail without sacrificing the coverage of long distance interaction. And this is what BootTHiC 2 aims to do. BootTHiC 2 is a form of hierarchical clustering which uses a network representation of the HiC data where the main purpose is to perform a graph cut of that interaction network, and thus separating each network into two partitions that are kind of preferentially self-interacting. The main feature of BootTHiC 2 is that at every iteration it evaluates what is the best resolution at which to perform the cut by maximizing the level of detail and the level of coverage of long-range interaction. This process of hierarchical clustering, where we apply recursively this spectral clustering, produces a sort of hierarchical tree cluster. Typically on a run of BootTHiC, we obtain around 500,000 clusters per genome. Among them, we are especially interested in those clusters that are enriched in transcriptional activity. And here transcriptional activity needs to be understood as the number of transcriptional events we can detect within a cluster. And so what we are wanting to do is to find whether or not some clusters have an exceptionally high count of transcriptional events within them. And to do this estimation or evaluation, we compare this observed count with a randomized count we obtain through bootstrapping. And here the bootstrapping is somewhat more involved than simply shuffling the peak coordinates of the cage transcription start sites. We try to reflect in the shuffling of those particular peak coordinates the sort of positional bias that the cage peaks tend to have with respect to gene bodies, their tendency to be closer to promoters and avoid intergenic regions and such things. We are able to do this by using chip seeker annotation of those cage peaks for each sample, and then try to reproduce the sort of annotation composition that chip seeker produces with the shuffled coordinates that we produce for each bootstrap. This essentially produces an empirical p-value that we then correct for multiple testing by using a method called dagger that accounts for the nested dependency between the clusters and the count that we produce. Typically, on a run of BootTHiC over a whole genome, we tend to find around 1,000 hubs per genome. This collection of hubs was produced for 3 human cell lines, H1, HMEC, and GM12878. Let me now try to present to you some of the outstanding results for those hubs. But first, let me give you some intuition as to how those hubs look like. And here I present to you the hubs that were produced for chromosome 19 for all 3 cell lines on which we ran the algorithm. And the thing I would like you to focus on are 2 aspects of those clusters. The first one is their very different sizes across cell lines, and also their tendency to, despite let's say mobilizing similar regions along the chromosome, the neighborhood that is produced for different cell lines tends to be very different. So that is indicating that despite maybe involving similar chromosome regions, the context in which those regions are actually existing can be quite different across cell lines. Now, the sets of results that we present to you in order to evaluate the functionality of those hubs revolve around this notion of whether or not being inside a hub is in any way exceptional. And we estimate this using 2 facets of the content for the hubs we're able to detect. One focuses on gene content and especially the transcription start site specificity, and the other set of results focuses on the enhancer content, especially in terms of the activity of those enhancers. Looking at the gene content of those transcriptional hubs, we wanted to evaluate whether or not this particular gene content had any specialization, and whether this specialization was different when comparing transcription start sites and genes inside the hubs compared to genes outside the hubs. And the way we did this is by simply applying a gene ontology enrichment analysis, where for the background we considered all the genes associated with active cage peaks, as annotated by Phantom 5, and in the foreground we considered all the active genes, but that were specifically found inside the hubs. The enrichment was estimated using a hypergeometric test and followed by an FDR p-value correction. What I'd like you to observe here is that for all three cell lines, there are very specific types of gene sets that seem to be highlighted and enriched in those particular hubs. In the case of GM12878, which is an immune cell line, we tend to find an enrichment in immune function. In the case of HMEC, which is an epithelial cell line, we tend to find an enrichment in adhesion, wounding, and epithelial function. And in the case of H1, which is a stem cell, we tend to find a sort of housekeeping enrichment, but also for some histone and nucleosome biology function. All in all, there's an indication that the gene content of the transcriptional hubs tends to be more specialized and more biologically related to the cell line and their biology in which they are found. Looking now at the enhancer activity, what we wanted to examine is whether or not the enhancer activity for the enhancers found inside the hubs is in any way different for the enhancer activity for the enhancers outside the hubs. And the way we measured activity of an enhancer is by looking at three different epigenomic proxies: histone modification, H3K27 acetylation, RNA polymerase II binding, and the transcriptional level as indicated by the cage signal itself. For each enhancer, we basically mapped or assigned the maximum level of either three epigenomic feature that was found within the corresponding interval. And to compare the distribution of enhancer activity inside versus outside the hub, we simply compare this activity distribution in terms of a Wilcox-Ranson test to evaluate the significance. Here I plot the distribution values for enhancer activity inside the hub in red and outside the hub in blue, and the more to the right a curve is, the more active this particular enhancer distribution tends to be. And what you can see is that for all three cell lines, the enhancer activity tends to be higher inside the hubs. We then try to see whether there was an association of the hubs with cancer. And here we understand cancer as an aberrant gene expression pattern compared to the healthy cell line. And the way we tried to examine this is by examining whether or not this particular gene expression pattern, this change in gene expression, is different when looking at genes that are inside the hubs compared to genes outside the hubs when looking at healthy versus cancer cell line. In this particular analysis, we focused on hubs that were detected in healthy breast epithelia, here HMEC, and compared this expression level with the expression level of corresponding breast cancer samples, MCF7 and MDA, and performed a differential gene expression analysis using DSEC2. What emerged from this analysis is illustrated in the following plots. Here on the left, I have the log-fold change distribution for genes that are found inside the hub in red and outside the hub in blue. What you can notice is that there's a clear enrichment for downregulated genes with negative log-fold change for genes that are inside the hubs when looking at the level of expression inside cancer samples. This particular enrichment in downregulated genes for genes inside the hubs is further supported by the enrichment in more significantly downregulated genes as indicated by the p-value distribution, which is enriched in lower p-value for downregulated genes inside the hubs. All in all, these results indicate a sort of shutdown of the genes that are originally found in healthy samples within transcriptional hubs. The take-home message I would like you to take is that BootTHiC is a method that detects candidate transcriptional hubs by integrating HiC and cage. It does so by focusing on clusters that are enriched both in chromatin interaction and transcriptional event. I have shown to you that these particular transcriptional hubs tend to be enriched in specialized genes, more active enhancers, and tends to be shut down in cancer. I would like now to thank my group for supporting this work and also my funding agencies, especially Scientia Fellowship. Thank you for your attention.